Okay, thank you again for the invitation. Big responsibility from your words. So I am a researcher. I work in the south of France. It has been a while that with my group we try to understand better neurological diseases, in particular Dravet syndrome. And today, I will talk about my work and the work of my colleagues, preclinical academic research. But preclinical research starts with the patients, and the inputs that we have from the clinicians, so the clinical evaluation, and that includes effective therapies, and from a geneticist, so the genetic evaluation of patients, the genetic variants that have been identified. And as basic researchers, we work with experimental models. So we try to generate models of the disease using different methods. Methods that can be simple cell culture, single cells in a dish. So in vitro models, they can be samples from the patients or more complex in vitro systems like these brain organoids that are 3D cultures. Basically, they mimic a structure that can be a brain structure of a human. And then we work with organisms like this fish that is a zebrafish or rat mouse models. But we try to understand pathological mechanisms and identify therapeutic approaches that then should be applied, translated to patients. And the first thing you have in mind in general when there is this translation is about novel drugs. Novel drugs with new clinical trials. But this is not the only thing that we translate. We translate also other things. For example, we can find how to use established drugs for new diseases. This is called drug repurposing. So a drug that is used for another disease that we find useful for the disease we are studying. Then we can improve the knowledge about the disease, identifying pathological mechanisms. And this is not just knowledge, basic knowledge, because these pathological mechanisms can be new drug targets. Then we can help and improve diagnosis of patients and also improve the selection of available therapies, because often clinicians have a lot of different drugs that can choose to treat a patient. But it's difficult to find the best one. And the preclinical research can help with that. And so, in general, all this is a precision medicine framework in which basic research, preclinical research is important. Now, something about the pathological mechanisms, so the causes of the disease. When we talk about Dravet syndrome, most of the patients have a genetic variant, a mutation, in a gene that is called SCN1A. The gene produces a protein that is called NAV1.1, and this protein is dysfunctional because there is the genetic mutation. This protein, NAV1.1, is a voltage-gated sodium channel that generates an electrical signal, a current, that is important for generating the electrical activity of the brain, of the neurons, the action potentials of the neurons. But one of the simplest experimental models that we can use is a culture, is a 2D culture of cell lines that don't express this protein, NAV1.1. and we make them express the protein, the human protein. And so we can record the activity of the protein with electrophysiological methods. So we can record the current, the electrical activity of these proteins, of these channels. And so we record something that we call sodium current here. And then this is the normal protein, normal NAV1.1 protein. When we express the mutant protein, mutant NAV1.1, we can see dysfunctions. For example, in a Dravet syndrome mutant, we can see a decrease in this current. You can compare the two here. You see that this is smaller. This is a loss of function that is typical of Dravet syndrome. And in other cases, we can see larger currents. So this is a gain of function. And this is something that we see in a different disease that is caused always by mutations, the genetic variants in the same gene, SCN1A. But in this case, with the gain of function, we have a different phenotype, a different disease that is called early onset developmental epileptic encephalopathy. And when we have a gain of function, differently than Dravet syndrome, drugs that are called the sodium channel blockers that reduce this current can be used. These drugs are usually not used for Dravet syndrome. And so these experiments, these recordings, are important for diagnosis because we see a gain of function. So we know, for example, this is not a loss of function typical of Dravet syndrome, and also for the selection of therapies because we can select drugs that reduce the activity of NAV1.1 that we don't use for Dravet syndrome. So this is a simple experimental system, but it can give you important information. This is what we have done in these papers, for example, with a lot of colleagues, in which we have identified this gain of function mutations. Then in vitro system, a little bit more complex, can be cultures of human neurons, in particular human neurons, generated from skin biopsy from the patient. So a piece of skin of a patient can be converted into neurons. And so we can study with this system the neurons of the patient. And a similar method can be used to generate organoids. So these are tissues, basically. In this case, we have in a dish a structure that can be similar to the brain cortex, for example, formed by human neurons. And so we can study function and dysfunctions of the neurons there. Then these are I showed you, are simple in vitro systems. Then we can use also organisms. For example, as I told you, there are fish can be used as a model of disease. And these fish that are called the zebrafish are useful because you can do high throughput screens of drugs. So you can test a lot of drugs with this model. But of course, fish are less similar to humans than mammalians. Mammalians like mice are more similar. They have a behavior that is more similar to our behavior. So we can study the epilepsy there in mice, and also behaviors that are similar to the behavior of humans. And here I will tell you something about pathological mechanisms that we have investigated in mice. This is the first mouse model of Dravet syndrome that we generated a long time ago. We introduced in the mice the same mutations that the geneticists found in a Dravet syndrome patient. And this is a truncation, so a loss of function mutation. You don't have any more function of the NAV1.1 protein. And the mice reproduce the phenotype of the patients. So the mice, the heterozygous mice, like the patients, have epileptic seizures. You can see here a generalized tonic-clonic seizure that you can always also record with the EEG here, like in patients. And so these mice have a normal development. They start to show hyperthermia-induced seizures 15 days of age. Then they show major spontaneous tonic-clonic seizures after P18, so they think they life, like the one you saw here. They have increased mortality because of sudden unexpected death in epilepsy, and they have cognitive and behavioral deficits. So they reproduce well the phenotype of Dravet syndrome patients. And something that you cannot do with patients, we have done with the mice. So we extracted the mice from the mice the neurons of the brain, and we recorded sodium currents in these neurons. And we found here a big reduction in sodium currents in GABAergic neurons, that are the neurons that inhibit the brain networks. And we also found a reduction in the excitability, so in the electrical activity of these neurons. You see here the normal neurons that have all these activities, so these action potentials, and they are reduced a lot when you have the mutation. So you have reduction of the sodium current, the excitability, and these inhibitory GABAergic neurons. And so this is basically the mechanism we discovered for Dravet syndrome in these mice. You have a loss of function because of the genetic variant of NaV1.1. You have reduced excitability of these GABAergic neurons, and because of that, a reduction of the inhibition on the network. So you have less inhibition, more excitation, so the brain networks are more excited, and this is consistent with an epileptic phenotype, so hyperexcitability of the brain. And this finding has been confirmed by a lot of colleagues. These are just few scientific articles that confirm this result. So this is the pathological mechanism of Dravet syndrome identified in a mouse model. Now, about therapies, you can use mouse models also to develop therapy. And Anna already told you about Stoke Therapeutics. This company developed a drug that is a little piece of DNA, an antisense oligonucleotide, or ASO, to treat Dravet syndrome, because this little piece of DNA can increase the expression of NaV1.1. Because you have a reduction in the function of NaV1.1, if you have more NaV1.1, you can reduce the severity of the disease or cure maybe patients. And so this drug is in a phase 3 clinical trial that is called Emperor, as Anna told you. And the evaluation and validation of this therapy in a mouse model of Dravet syndrome has allowed the clinical trial, because the clinical trial has been started after the successful study in mice. This is the scientific article that reported that study from the laboratory of Lori Eisen. Here they injected into the brain the drug before the onset of symptoms in the mice. And here you have a quantification of the mortality of the mice. So this is the control, the mice that don't have the mutation, that don't die. These are the mice with the mutation in red that die. So this is not like in patients. Mice die much more than patients. You have here 40% mortality at 90 days of age. That is a lot. You don't have the same thing in patients. This is typical of mice. So you basically can quantify the phenotype severity of the phenotype of the mice, quantifying the mortality. And you can see here that when you treat the mice, you reduce a lot the mortality. It's this blue line here from basically 40% mortality. You have here, after the treatment, less than 20% mortality. So this is a very good effect of the treatment. One thing I want to highlight is that the effect that you have is mainly at young age, because if you look at the mortality at later age, when the mice are adult, you have similar mortality. You can compare the curves here, the red one and the blue one, they are similar. You reduce a lot of the mortality when the mice are young here. Okay, so this is an example. This is an important example because these experiments allowed basically the first clinical trial of this drug. There are a lot of therapeutic approaches that are evaluated in mouse models and other models by colleagues. I don't want to give you a list here. This is just an example. I want to highlight the limits of this study because but it's a very important study, but there were features that were not evaluated that are important. So sex differences, difference between males and females here in the mice were not evaluated. The treatment was just before the onset of the symptoms. The scientific studies should treat also adults, so at later ages. And here the effect on behavioral cognitive functions was not investigated, only the effect on seizures and mortality. And so these are important features that have to be included in current and future evaluation of therapies in preclinical studies. So again, about here, more pathological mechanisms than therapies. This is a study that Anna mentioned already to tell you that if you rescue NAV1.1 also late during the disease, also in adults, you can improve a lot the symptoms. So this is a study done by the laboratory of Vanya Brockman and Gaia Colassante, and they reactivated the gene. Basically, they started with mice that had the mutation, and then they removed the mutation. So this is something you cannot do in patients, so it's not a therapeutic approach because it's too complicated to do it in patients. You can do it in mice. And so you can basically remove the mutation, and you have mice that initially had the mutation, and then they don't have any more the mutation. And when you remove the mutation, you remove the seizures, basically. This is the control. You see here the bars. Each bar is a seizure. Each line is a mouse. They have a lot of seizures. Also, when they are adults, so these are 90 days old. When you reactivate the gene, so you remove basically the mutation, you don't see any more seizures. So basically, you cure completely mice that had a Dravet syndrome phenotype. You cure them when you remove the mutation in adult mice. So also treatment in adults can be effective. Now, back to the therapies and also the pathogenic mechanisms. So I told you that there is an initial cause here, that is the pathogenic mutation in the gene SCN1A. You have an initial functional effect. So NAV1.1 loss of function, decrease sodium current, and decrease excitability of these GABAergic neurons that are important for the inhibition of the activity of the neuronal network. And I showed you therapies that reduce the dysfunction, this initial cause. But when you have a modification in a biological system, the biological system, so the organism, responds to the modification. And what you see at the beginning is just the tip of the iceberg, because then you have secondary responses. Secondary responses that can be pro-pathologic, so they are modification in the features of the cells of the organism. In some cases, they are pro-pathologic because they amplify the initial dysfunction. In other cases, they are homeostatic because they tend to reduce the initial dysfunction. And then the mature phenotype, so the mature disease, is caused by the sum of all these things. So the initial dysfunction, the pro-pathologic secondary responses, and the homeostatics secondary responses. And this is complicated. So you start with something that is relatively simple, a single mutation in a single gene. And then you have something that is much more complicated with all these secondary responses. But this can provide also opportunities, because these secondary responses can be targets for therapies. So you can design a therapy to reduce a pro-pathologic response, or also to increase an homeostatic response. And so you have more choice in your therapeutic strategy. But I want to tell you a few words about something we did to increase an homeostatic response. That is part here on the right of the screen. So we identified an homeostatic response in a mouse model. So we identified some GABAergic neurons that are more active than the controls. This is the opposite than we expected. And we identified it as an homeostatic response because these neurons that are called CCK, because they release cholecystokinin, that is a neuromodulator, implement an homeostatic response. So they release more cholecystokinin, and cholecystokinin can increase the activity of other GABAergic neurons that, as I told you, are less active in Dravet syndrome. So cholecystokinin produced by the CCK positive neurons can help the other GABAergic neurons in increasing their activity. And so this is an homeostatic response. And so we used cholecystokinin as a treatment. So we administered cholecystokinin with intranasal administration to amplify this natural homeostatic response. And this therapeutic approach worked because when we administered cholecystokinin to the Dravet syndrome mice, we decreased the seizure frequency and severity, and we improved sociability and memory, so cognition. So this is one of the first therapies that targets an homeostatic response, trying to amplify it. So I told you that this homeostatic response are important, and that also other secondary responses can be targets for therapies, but we have to identify them. And there are now novel techniques that can be used to identify the secondary responses. Some of them are called transcriptomic techniques, like this spatial transcriptomics that I'm showing here. This is a slice of a brain of a mouse. And the colors that you see, each dot there is a single neuron. Basically, you can identify the properties at the level of gene expression of each neuron in the brain of this mouse. And you can identify things that are modified by the secondary responses. And another technique that we start to use to better understand pathological mechanisms is the recording of neuronal activity with, for example, this technique that is called the neuropixels that allows to record with thousands of electrons. Basically, you record here the activity of thousands of neurons in the brain of a mouse. And you can identify dysfunctions there. And you can correlate the dysfunctions that you have in the activity of the neurons that you record with these neuropixels with the dysfunction that you have in the gene expression in these cells. And so you can better identify secondary responses that can then be targeted for therapies. So my conclusions are here. I told you that we have a lot of different experimental models that are useful not only for screenings of new drugs, identification of new drugs, but also to identify pathological mechanisms, to repurpose drugs, to use drugs for new disease, drugs that we know already, but that we can use for other diseases. Also select established drugs in a precision mechanism framework, so adapt the therapy to the single patient, etc. And so there are also numerous experimental models that can be used according to the objective that you have. You can study the dysfunctions of the proteins, the dysfunctions of the cells that express these proteins, the dysfunctions of the networks of cells, so the neurons in the brain, the dysfunctions of the organism, and then they can be used to screen the effect of drugs at low, medium, high throughput according to the experimental model that you use. There are novel experimental models that may be exploited in the future, like organoids, but more studies are necessary for validating these methods that are kind of new. And an important thing is that all the experimental models have limits because they are models. Each model can be used within certain limits. And so they have to be used and interpreted with caution. Then it is important to include in the study design, age of treatment, sex, and behavioral cognitive functions. In the past, this has not been done always. And there is an increased evidence that secondary responses are important in the development of the disease. They should be better studied with novel techniques that some of them I try to highlight in my presentation. And these secondary responses can be targets for therapeutic approaches. And then I thank you for your attention. I thank you also my collaborator. And this is just a beautiful picture of the south of France. Thank you.